In this session, we are going to learn about the anatomy of dicot and monocot leaf. So let us start with the anatomy of a dicot leaf first. So the dicot leaf is also called dorsiventral leaf. Dorsiventral leaf. Alright, so if we look at the vertical section of a dicot leaf, then it shows three main regions or three main parts. The first is the epidermis, which is present both at the upper and the lower surface of the leaf. Then we will find mesophyll cells and vascular bundles. So we will be studying primarily about these three parts when we talk about the anatomy of dicot or even in monocot leaf. Alright, so if we take the vertical section, these three parts will be visible: epidermis, mesophyll and vascular bundle. So now let us understand the anatomy with the help of the diagram. We will start with the dicot leaf first. So first of all, we will see that the upper layer is epidermis. The leaf actually has two layers, one upper layer and one lower layer. So both the layers will have epidermis. The upper epidermis is also called as adaxial epidermis and the lower epidermis is also called as abaxial epidermis. Now both of these epidermis will have a conspicuous layer of cuticle. We already know the function of cuticle, that it prevents water loss, and this cuticle is usually present in the stems and leaves because water is lost in the form of transpiration. So in order to prevent the water loss, the cuticle will be present. So a layer of cuticle will be present both at the adaxial and abaxial surface. It is a conspicuous layer. Now below the epidermis, we will find a tissue, a special type of tissue which is found in the leaves, and this tissue is called mesophyll. It covers the whole of the internal region of the leaf. Alright, the cells of these mesophyll, they have chloroplast, and hence they carry out the function of photosynthesis. We already know that leaves are the primary organs which carry out photosynthesis, right? So it is these mesophyll cells or the mesophyll tissue inside the leaves which have chloroplast and they carry out photosynthesis. So if we talk about the mesophyll in dicot leaf, there will be two types of mesophyll tissue we will see, and these mesophylls are made up of parenchyma cells. Alright, so let us see the two different types of mesophyll which we will find. So below the upper epidermis or the adaxial surface, we will find elongated type of mesophyll cells like this. These will be arranged vertically and parallel to each other. In this manner. Alright. So these type of mesophyll cells are called palisade mesophyll or palisade parenchyma. These palisade parenchyma are vertically arranged elongated cells. They are present parallel to each other, found only at the adaxial surface. And now at the abaxial epidermis or on the lower epidermis, we will see round and oval spongy parenchyma or spongy mesophyll, which is the second type of mesophyll cell. So these are round and oval, and they have large number of cavities or intercellular spaces between them. They are not as tightly packed as palisade parenchyma. They are very loosely arranged. These are called spongy mesophyll, found at the lower epidermis, towards the lower epidermis. Alright, so these palisade and spongy mesophyll constitutes the two types of mesophylls present in a dicot leaf. Now we are done with the epidermis and mesophyll. Now let us draw what is remaining, that is the vascular bundle. So vascular bundles will be found wherever there is a vein in the leaf, right? So suppose here one vascular bundle is present. The vascular bundle will have xylem and phloem inside it. So suppose these are xylem, and then there will be phloem for conduction of water and food. And this vascular bundle will be covered by cells which forms the bundle sheath. So they are quite thick. These cells are quite thick and they form a layer of covering around the vascular bundle, and this is called bundle sheath. Bundle sheath because it is covering the vascular bundle, hence it is called bundle sheath. Alright, inside the bundle sheath there will be vascular bundles, xylem and phloem. And these vascular bundles will be present whenever there is a leaf, a vein. Whenever there is a vein or veinlets inside the leaf, there we will find the vascular bundles. Now, the stomata, if we talk about the stomata, then the adaxial or the upper epidermis may have stomata or may completely not have stomata, but stomata is usually present at the abaxial epidermis. Alright, so this is one stoma. This is one stoma. Stoma is the singular of stomata. So here is the stomata. We all know what is stomata. We have studied about it in our previous lectures. So stomata is from where the gaseous exchange takes place in the leaves, right? So this stoma is internally connected to these air spaces between the spongy mesophyll, these air cavities. Hence the gaseous exchange takes place between these air cavities inside the leaf to the outer atmosphere. So these are the substomatal cavity. Alright, and we have air spaces here between the spongy mesophyll. Yeah, so this was all about the anatomy of dicot leaf. Alright, so the three main parts were the epidermis, mesophyll and the vascular bundle. We saw that the epidermis was present on both the surface of the leaf, when we talk about the dicot or dorsiventral leaf, the upper epidermis is also called adaxial epidermis and the lower is called the abaxial. Alright, both the surface of the epidermis will have a continuous layer of cuticle to prevent water loss. The upper surface of the epidermis may or may not have stomata, but the abaxial epidermis usually has stomata through which the gaseous exchange takes place. And inside the epidermis, the whole of the leaf is covered or filled with mesophyll tissue. This mesophyll tissue contains chloroplasts that carry out photosynthesis and is usually of two types, palisade and spongy mesophyll. Palisade mesophyll is found at the upper epidermis and is made up of vertically arranged and parallelly arranged elongated cells. Then towards the abaxial epidermis we have round and oval large spongy mesophyll which have a large number of air cavities between them. These cavities are connected to the stomata for gaseous exchange, and both of these type of mesophyll contains chloroplast and carry out photosynthesis. Then whenever there will be veins or midribs inside the leaf, usually at those places we can find the vascular bundles. The vascular bundle is covered by thick walled cells which are called bundle sheath. Alright, and if you remember when we studied about the types of venation, there we studied that the dicot leaves have reticulate venation, and in reticulate venation the thickness of the veins vary. Alright, so the size of these vascular bundles found inside dicot leaf will depend on the size of the veins of those veinlets. Usually the main vein or the midrib is the largest vein, so the vascular bundle of that vein would be the largest, and depending on the size of the other veins the size will vary. Alright, so size of vascular bundles vary depending on veins and veinlets. So this was all about dorsiventral or the dicot leaf anatomy. Now let us move on to the monocot leaf or the isobilateral leaf. Anatomy of monocot or isobilateral leaf. So here isobilateral means what? It means both of the surfaces will be of similar type or the same type. So as we studied that the leaves have two surfaces, and in case of dicot leaf the stomata were present more towards the lower epidermis, and the upper epidermis may or may not have stomata. But in case of monocot, the stomata are present at both the surfaces of the leaf, both the upper and the lower surfaces. Alright, so let us draw the structure or the anatomy. This will be epidermis, adaxial. Now I will draw some of the cells as large cells because these are the special type of cells which we will study later. So this is adaxial epidermis and this one is abaxial. I'll have to make space for the stomata. Let us draw one stomata here. So the stomata will be present on both the surfaces, upper and lower. Now the structure is quite similar to that of dicot leaf. The only difference is that the mesophyll will not be differentiated into palisade and spongy mesophyll here in case of isobilateral leaf. Otherwise, most of the structure is similar. Alright, so there will be adaxial and abaxial epidermis. So the first difference is the stomata will be present on both the sides, adaxial and abaxial epidermis. So here one stoma, here another stoma. The mesophyll will be only of one type. There will be no differentiation into palisade or spongy. So let us make mesophyll cells. Also, the epidermis will have a layer of cuticle to prevent water loss. Then we will have vascular bundle, xylem and phloem. So suppose this is xylem, this is phloem. Bundle sheath. Okay, so I'm not labeling these parts because they are quite similar to that of dicot leaf. Now let us come to the special part or the unique feature of the monocots. Now in case of grasses, certain cells of the epidermis, usually which are closer to the vascular bundles, if this is the vascular bundle, then there are certain epidermal cells which are situated closer to the vascular bundles. They get enlarged. In case of grasses, these epidermal cells get enlarged and they become colorless and empty, and these are called bulliform cells. Let me use a different color. So this cell that I drew in the epidermis, the large ones. So these special cells are large and empty and colorless cells, usually found at the epidermis of the grasses, and they are called bulliform cells. Alright, so these bulliform cells are found close to the vascular bundles, and what do they perform? What happens is whenever the leaves absorb water, then these cells become turgid, and once they are turgid, they help the grasses to expand their surface. Alright, and whenever there is a loss of water, whenever there is a stress, water stress or scarcity of water, in that case these bulliform cells become flaccid, and when they get flaccid or sunken, then the leaves curl their surfaces in order to prevent water loss. So that is the function of bulliform cells. I hope it is clear to you. Bulliform cells are special epidermal cells which are large, colorless and empty cells. They absorb water and become turgid. Once they are turgid, the surface of the leaf expands. Alright, and whenever there's a scarcity of water, they become flaccid and the surface of the leaf curl inwards in order to prevent water loss, and these are only found in grasses. I hope it is clear to you. And these bulliform cells are only present at the adaxial epidermis. Alright, that's why I have drawn them here on the adaxial or the upper epidermis. So these bulliform cells will be present at the upper epidermis of the grasses. Please remember this. And if we talk about the venation in monocot leaf, then you must remember, if you have gone through the previous lecture, that the monocots have parallel venation, right? And in parallel venation, the size of all the vein and veinlets are quite similar. The only exception is the main vein. Leaving the main vein, all the other small veins and veinlets are of equally similar size, and therefore the size of the vascular bundle is almost similar in case of monocot leaves. Alright, whereas in case of dicot we saw that because of the reticulate venation, the size of the vascular bundle vary, and therefore size of the veins and veinlets vary, and therefore the size of the vascular bundle vary. Whereas in case of monocots, the size is almost similar. Alright, so I hope the anatomy of monocot and dicot leaf is clear to you, along with the unique differential points. The structure would be similar in case of dicot and monocot leaf anatomy. All the structures and the parts would be similar, except that in case of monocots, in case of monocot leaf, the stomata will be present equally on both the surfaces of epidermis, and mesophyll will not be differentiated into palisade and spongy parenchyma. The size of the vascular bundles are almost similar or the same, and besides that, the adaxial epidermis have bulliform cells, and I mentioned the function of bulliform cells. So I hope everything is clear to you. That's all for this lecture. Thank you so much.